Hi, my name is Ayesha, and I am a second year medical student at the University of Calgary. I've also completed a microbiology degree here. Today, I'm going to be talking about the COVID-19 virus, specifically how it infects us, then how our immune system protects us, and finally, a little bit on how the vaccine works. So without a further ado, let's get started. Let's start with the COVID-19 virus molecule. The COVID-19 virus is a small shell-like molecule coated with tiny non-toxic spikes. Let's call them spike proteins. These spike proteins will become very important in a few minutes, but before we get ahead of ourselves, let's talk about what's inside of the virus. The inside of the virus is hollow, and it contains a thin string-like strand called RNA. RNA is the genetic code of the virus, so it contains the virus's genes, just like our DNA contains our genes. So how does this molecule infect us? Firstly, you would have to be exposed to airborne droplets from an infected person. If these virus-containing droplets enter our body, the virus is inside. Now that it is inside us, it searches for cells to infect. Once it finds a cell, the virus spike protein acts like a key to enter the cell. Then the virus starts replicating. These new viral-like particles then leave the cell and go on to infect other cells. So how does our body protect us? As our immune cells encounter these viral molecules, they slowly start recognizing that they are abnormal. Our immune cells then memorize the spike proteins and tell the rest of the immune system, hey, anything with the spike protein is bad and we have to kill it. Our immune system then produces molecules called antibodies. These have two purposes. One, to latch onto the spike proteins, preventing the virus from entering our cells, and two, to act as kill signals for our immune system. And that is how our immune system efficiently protects us. In the future, if our body is ever reinfected with COVID-19, our immune system won't be as slow as it was the first time. Remember, it already has the spike proteins memorized, so it'll employ the antibodies and kill the virus off quickly. Let's discuss the vaccine now. You can think of the vaccine syringe as an olive jar. In an olive jar, you have olives as well as fluid that is preserving the olives. Similarly, the vaccine syringe contains a biological agent, which is the protective ingredient, as well as preservative fluid. Sometimes the preservative fluid can cause allergic reactions, but that's not always the case, and that's why you're asked to wait 15 minutes after you've been vaccinated. So how does the vaccine's biological agent protect us? In COVID-19 vaccines, this agent contains instructions for the non-toxic spike protein. These instructions will tell your cell to produce loads of COVID spike proteins. Your immune system will recognize these spike proteins as abnormal, memorize them, create antibodies, and destroy them, just like it would in an actual COVID-19 infection. That is why you might feel a little feverish or sore after getting the vaccine. One important thing to note is that the vaccine actually prepares your immune system more than a COVID-19 infection would. So basically, if you take the vaccine, you'll have stronger protection. Also, keep in mind that the structure of instructions will differ between vaccine brands. For example, Pfizer and Moderna contain an mRNA gene for the non-toxic spike protein. Regardless of the vaccine, though, your body will eventually digest these instructions. Finally, let's discuss variants and vaccine booster shots. A COVID-19 variant is made when the spike protein mutates. Vaccine brands are currently adjusting their instructions in their biological agents to provide protection against different variants. They will administer these updates through booster shots. And that brings this portion of the presentation to a close. Thank you for joining me in learning a little bit about COVID-19.
Hi, my name is Fizah. I'm a clinical researcher and an instructor for health administrative studies. I have studied biology and biomedical technology from the University of Calgary. So today I'll be talking to you for a few minutes. I'll be talking about the COVID-19 vaccine and the clinical trials or clinical research that went behind manufacturing this vaccine in a way that is safe, efficient, reliable, and transparent. I just mentioned the terms clinical trials and clinical research. Why are they important to us in the COVID-19 pandemic? Let's talk about it a little. Clinical trials is a type of research where researchers evaluate surgical, medicinal, or behavioral interventions. These are also called experimental treatments. A clinical trial is used to learn if a new treatment is safe, effective in humans. It's also used to learn about the dosage and the side effects of these new treatments. Simple terms, it's basically doing experiments in human beings. If that's the case, then how do we know they're safe? How do we know if there are any quality control measures to assure safety and accuracy? What about the transparency? And what about COVID-19 vaccine trials? To understand that a little, it's important to know that clinical research is divided into several phases. Before I explain these phases to you, I want us to talk about the reliability of these clinical trials. Let's imagine a staircase with a destination at the end. The destination for clinical trials means to be able to provide this experimental treatment to the general public. These phases gradually grow confidence in the experimental treatment. These stages also assure that checkpoints are present before moving on to the next phase. So let's talk about these checkpoints. Prior to the start of any study, the study design is submitted to national competent authorities and ethics committees. Once evaluated and approved, only then a study can begin. In the case of COVID-19 vaccines, all experimental vaccines that are approved are randomized and blinded. What that means is neither the participant nor the treating doctor knows who is being assigned the treatment or who is being assigned placebo. Placebo is something that doesn't actually contain the treatment, but looks like the treatment. So what does it do? It avoids bias that could result in heightened results and heightened estimation, side effects or benefits. The next one is safety run-ins or checkpoints. These are performed within each phase or after the phase of the trial. The entire study is evaluated, and this is what allows us to know whether or not to move on. Last but not least, resubmission is required when a change is made to a study. A change could be adjusting the dose, changing the population that is eligible, or changing the treatment regimen. I hope this gives us some trust in clinical trials and the process involved. Now let's talk about the phases. The first phase is called preclinical research and development. It involves developing the treatment itself, and at this point, the treatment is not taking place in humans. The next phase is phase one. Phase one takes place in a very small population. Phase two happens in a bigger population, and now we're gathering more and more data. Phase three then, these are large trials that help us learn more about the experimental treatment. That's because phase three trials happen in an even bigger population. Now we're talking about up to 3,000 participants. Finally, phase four. Now this is when a treatment is approved and available across pharmacies and hospitals for general public. The data that is gathered from this phase allows us to study long-term benefits and side effects of a new treatment. This sounds like a lengthy process. Then how was COVID-19 vaccine developed so fast? Let's break that down a little. The first factor is the level of emergency. I think not much needs to be explained about a pandemic that's costing us millions of lives. The next factor is funding. An average clinical trial costs more than tens of millions to hundreds of millions of dollars to complete. With large funds that were allocated towards the development of COVID-19 vaccine, researchers were able to avoid the time that's spent collecting funds. Next one's perhaps the most important one. We might think that the recipe for COVID-19 vaccine was dropped from heavens last year, but that's simply not what happened. The basic research on DNA vaccine began at least 25 years ago, and RNA vaccines have benefited from 10 to 15 years of strong research. In addition, vaccine for similar viruses such as SARS has been developed before. What does it mean for us? This basically means we had the recipe, and this allowed us to swap the ingredients with COVID-19 particles to develop safe and effective vaccines fast. Last factor is global collaboration. The size and shape of COVID-19 virus was made to be a public knowledge. With this information, vaccine development companies didn't have to spend the extra time to find out how COVID-19 works. Everything that I mentioned takes months to years to complete. Subtracting this time allowed the COVID-19 vaccine to be developed not only fast, but to also be safe and effective. Now, to end our talk, let's talk about the COVID-19 vaccines that are approved by Health Canada. These names might sound familiar to you. Now, what I have done is I have created a table to summarize brief details. My apologies, the last header reads side effects. I want to emphasize 3 points, but please do spend time on your own to study this table. The first point is the efficacy or percent effectiveness of these vaccines. These vary, and that's because averages are taken from a wide range. For example, let's look at AstraZeneca. You will see that the effectiveness ranges from 47 to 94%. The variation in this range depends on many factors, such as age and previously present medical conditions. I want you to also notice this. When you Google search how often you get blood clots from COVID-19 vaccines, you will get different percentages. And that's because this all depends on the eligibility criteria of the region that these percentages are being reported from. As an example, if you take data that comes from a region that only allows its elderly population to be vaccinated, it will look dramatically different than the percentages that are reported from a region that's allowing its entire adult population to be vaccinated. Third point, while it might seem like some vaccines work better and some don't work as well, it's important to notice that the hospitalization due to COVID-19 infections in patients who are vaccinated is practically zero in AstraZeneca and Johnson & Johnson. All right, now that we know that, let's talk about AstraZeneca's COVID-19 vaccine and blood clots. So I want you to note this. Granted, there's a risk of blood clots, but what's important to note is that the absolute risk for blood clot to occur after taking the AstraZeneca's COVID-19 vaccine is extremely low. But the risk of blood clots from the COVID-19 infection is more than 14,000 times higher. So what does that mean for us? The takeaway point is that the process that leads to vaccines being developed and manufactured for general public is highly regulated. That means it's very reliable. So to summarize, the benefits far outweigh the risks. I hope this video allowed us to see the strict regulations and the decision-making that goes behind distributing and manufacturing these COVID-19 vaccines. Thank you for joining me.